Hello, this is a video on SARS-CoV-2. My name is Harsh Sharma. I have a BSc, MSc, PGCE, and I did a second BSc. Please read the disclaimer. You may like to stop the video while reading the disclaimer. When you have read it, you may like to continue to watch the video. Please note, no advice is being given. It is not intended to give any advice. All information is given for educational purposes only. And also please note, I am not a medically qualified doctor. This presentation is of literature search on SARS-CoV-2 and search on other sources such as the internet and my interpretation thereof. It is limited as it has not been peer-reviewed nor has been subjected to expert opinion. I do not consider myself an expert on the issue. Indeed, this has been a learning experience for me. Furthermore, scientific ideas can change and information become out of date. We urgently need data generated from SARS-CoV-2, such as the virus structure, RNA replication, and so on. So we need data generated by experiments on SARS-CoV-2. I see many diagrams of SARS-CoV-2 on the internet which show wrongly that SARS-CoV-2 has hemagglutinin esterase. There is no clear statement whether or not M protein is linked to N protein. There is no clear statement on where structural proteins are synthesized, in the cytoplasm or ER. Yes, they eventually end up in the ER in any case. So we need fundamental experiments carried out on SARS-CoV-2 and data generated on SARS-CoV-2 itself. Name of the virus is SARS-CoV-2, Severe Acute Respiratory Syndrome Coronavirus 2. And the disease this virus causes is called COVID-19, coronavirus disease 19. Some authors use the name 2019 nCoV before SARS-CoV-2 was designated as name for this virus. Hence, 2019 nCoV and SARS-CoV-2 are two different names given to the same virus. SARS-CoV-2 is now known as the coronavirus. General public and popular press now use the term coronavirus as synonym for SARS-CoV-2. Coronaviruses are a large family of viruses that usually cause mild to moderate upper respiratory tract illnesses like the common cold. However, 3 new coronaviruses have emerged from animal reservoirs over the past 2 decades to cause serious and widespread illness and death. This is the general classification of viruses, the order Nidovirale, family Coronaviridae, and subfamily Coronavirinae. When we talk about coronaviruses, we are talking about Coronavirinae. So the Coronavirinae are the viruses generally known as coronaviruses. SARS-CoV-2 is a beta coronavirus. Within the Coronavirinae, there are 4 genus: alpha, beta, delta, gamma. SARS-CoV-2 is a beta coronavirus, and then in the genus beta coronavirus, it is a B coronavirus. Baltimore classification is another way of classifying viruses. The important thing about Baltimore classification is it gives you information about the virus straight away. So, viruses, if they have no reverse transcriptase, it's an RNA virus, a single-strand RNA virus, and it's a positive single-strand RNA virus, that is SARS-CoV-2. So SARS-CoV-2 does not have reverse transcriptase. It is a RNA virus, single RNA virus. The RNA is a positive strand. Therefore, SARS-CoV-2 is a class 4 virus according to the Baltimore classification. Coronaviruses are enveloped RNA viruses. Envelope is a lipid bilayer with viral structured proteins in it. RNA is positive sense. RNA is very large for a virus, 29.9 kb. For RNA virus, this is quite a large genome. And coronaviruses are widely distributed in mammals and birds. This is a general structure of a coronavirus. This structure is actually mouse hepatitis virus. This is a reference. I shall be using this structure to illustrate general structure of coronaviruses and trying to relate that to the SARS-CoV-2 virus. Would be nice to have a diagram of a SARS-CoV-2 virus based on research on SARS-CoV-2. Maybe I should have looked harder for such a diagram. There are many diagrams, but I really wanted a diagram which was created after research on SARS-CoV-2. Maybe I should have looked harder for such a diagram, but for the meantime I'm going to be using mouse hepatitis virus as to illustrate the general structure of coronaviruses, and appreciating this actually is not precisely SARS-CoV-2 structure. So there is the ribonucleoprotein. It is RNA, viral RNA combined with N protein in the inside the lipid bilayer. In the lipid bilayer are embedded viral structural proteins. This is the spike protein. This is the membrane glycoprotein M protein. Envelope protein, this small one here, is the envelope protein. And notice that the membrane glycoprotein M is actually attached to N protein. Not every diagram shows this attachment. I do feel that this matter needs to be cleared for SARS-CoV-2. Notice SARS-CoV-2 does not possess hemagglutinin esterase. SARS-CoV-2 virus genome has been sequenced and there is no sequence coding for hemagglutinin esterase. So we need the data to show whether M protein is attached to N protein in SARS-CoV-2. My personal view is that M protein should be attached to N protein in SARS-CoV-2. Viral RNA has organized structure, and to maintain this structure there has to be interaction between viral RNA and membrane proteins. Viral RNA is not there just as a lump. We need data to confirm or refute this. The ribonucleoprotein, the RNA, viral RNA, combined with N protein, is contained within the lipid bilayer. The lipid bilayer is made of phospholipid bilayer. Phospholipid bilayer consists of molecules with hydrophilic end and hydrophobic tails. The hydrophilic ends align themselves, the molecules align themselves, so the hydrophilic ends are facing the water. Here the hydrophilic ends facing the water, and the hydrophobic tails are tucked on the inside. This is shown more clearly on this diagram with the cell membrane, with water on the outside of the cell, water on the inside of the cell, and the hydrophilic part of the molecule facing the water. Hydrophilic part of the molecule facing the water. Hydrophobic tails are tucked away from the water. Lipid bilayer requires water on the inside and outside to be in functional orientation. So to maintain this lipid bilayer here, we do need water on the outside and we need water on the inside. So the virus survives in droplets, outside the body that is. However, even when dried up, it will take some time for the lipid bilayer to lose its functionality. It seems virus can remain viable for relatively long periods, even when on dry surface. For completeness, we must also consider whether this virus can reconstitute if placed in a suitable medium after drying up. And for optimal conditions, the aqueous solution must be of correct osmolar strength. So what is this on the middle here? What is it? Well, a clue has been given in earlier slides. It can't be nothing, it can't be air, and in fact it needs to be something in particular. It needs to be a watery solution, aqueous solution, because the virus lipid bilayer requires water on the outside and water in the inside for it to be maintained. So this needs to be some kind of watery solution. It's probably this bit of the cell cytoplasm the virus took with it when it matured. These are the electron micrographs of the SARS-CoV-2. Please take note, electron micrographs are always black and white. So when we see color electron micrographs, these are color enhanced or false colored, done in Photoshop and so on. Nevertheless, in this particle you can see very clearly the spike protein sticking out, which give the virus its name, the coronavirus, like the sun's corona. Virus particles are 60 to 140 nanometers, pleomorphic. So what does pleomorphic mean? It means here we are, the virus particle is on the smaller virus particles, larger virus particles, roundish viral particles, more oval virus particles. So pleomorphic means it has different shapes and sizes. And this is a virus going in the lung. Red is the immunostaining of the virus, so it positively identifies SARS-CoV-2. Here it is, SARS-CoV-2 in red here. In the bronchial epithelium, again positively identified as SARS-CoV-2. Green is the ACE2 in the bronchial epithelium. Here it is, a specimen from a patient, a virus shown by electron microscopy in the lungs. Please note this virus has not been positively identified. There should be immunostaining, immunogold and so on, which particularly identify this as a SARS-CoV-2. Nevertheless, it does look like SARS-CoV-2. You can see the spike protein sticking out and so on, and the fact that it came from a patient with COVID-19, all pointing towards this is SARS-CoV-2. But to be precise, we need immunogold staining to show that this is SARS-CoV-2. This is intestinal organoid cultures, that is, say, parts intestine taken and cultured in petri dish. So this is growing in petri dish in vitro, and in this case white is the immunofluorescence showing virus, again showing coronavirus can grow in intestinal tissue, be it on the culture outside the body. This is actually intestinal tissue from the body, from a patient infected with SARS-CoV-2, showing red immunostaining, positively identifying coronavirus in the intestine. Here the green is viral RNA, and you can see lots of viral RNA which has lysed the cell. So cell lysis has taken place, producing this viral RNA outside the cell. Here it is, the cells showing these cells are ready to be lysed. These cells have already lysed. So this here showing the virus carried out cell lysis as part of this reproductive cycle. But up here, this does not seem to be cell lysis. This seems the virus is growing in the cells without lysing them. And not only that, virus is growing specifically on one end of the cell. If you look at it, brown is a virus on this side of the cell, no virus on this part. Here it is, brown on this part of the cell, no virus on that part. Please note, this is a patient specimen, autopsy specimen, and this is the alveolus. The alveolus has actually lost its correct architecture. The cells are no longer in the right position, in the right orientation. Nevertheless, it shows clearly the cells are infected with the virus but not lysed. Now we start looking at the SARS-CoV-2, the life cycle of this virus. The virus binds the cell surface receptor, which is the ACE2, and the virus uses its spike protein to bind ACE2. Once it's bound, the enzyme TMPRSS2 found in the cell membrane cleaves the spike protein, and then further events follow which lead to the fusion of the viral membrane and the cell membrane, and an opening is created through which RNA enters the cell. This RNA, that molecule, same molecule that virus brought into the cell, is then used for making proteins straight away. So not all of the RNA, but some of it is actually then utilized to produce two polypeptides. Each of these polypeptides is a multiprotein. It contains several proteins within it, and the proteolysis is carried out to produce individual proteins. Then these proteins are the known structural proteins that are used for viral RNA reproduction, host cell evasion, and so on. And one of the jobs they do is they induce the cell to make these double membrane vesicles. Double membrane vesicles are not normal structure in a cell. They're induced by the virus. Then the RNA and the proteins that are going to help copy the RNA, make RNA synthesis, move into the DMV, double membrane vesicles. From the plus strand, minus strand RNAs are produced in the DMV, and notice not just one single strand RNA is produced, several species of negative strand RNAs are produced, different lengths. Each of these different lengths of negative strands is copied to produce a positive strand of RNA. Then these come out of the DMV and are translated, proteins are synthesized, spike envelope and M glycoprotein, which then go to found in the endoplasmic reticulum. From there on, they move on to the endoplasmic reticulum Golgi intermediate compartment. The nucleocapsid, the nucleoprotein combines with the whole RNA to make nucleocapsid, which also moves into the ERGIC, the endoplasmic reticulum Golgi intermediate compartment, which then goes to the Golgi apparatus, which then goes on to make vesicles. In the vesicle, spike protein is cleaved by at another site, S1, SS2, by furin, and then the virus is released by exocytosis. Now we will discuss the virus entry into the cell in more detail. SARS-CoV-2 uses ACE2 as a docking site on the cell surface. So ACE2 is the receptor for the virus under normal conditions. Virus uses its spike protein to attach to ACE2 on the cell. SARS-CoV-2 enters the cell via membrane fusion or endocytosis. SARS-CoV-2 enters the cell via membrane fusion or endocytosis. This is the membrane fusion pathway. The virus attaches to cell surface receptor ACE2 via its protein, spike protein. The enzyme TMPRSS2 in the cell membrane cleaves the spike protein at S2, and events follow that leads to membrane fusion of the viral membrane, and the cell membrane fuse. Opening is created to allow the viral RNA into the cell. This is the endocytosis pathway. The virus attaches to the ACE receptor by spike protein, but we do not need TMPRSS2. There is no cleavage at S2 at this stage. The virus is then taken into the cell via the endocytic pathway. Here is the virus in an endosome. It is then released. The viral RNA is released from the virus via the action of enzyme cathepsin L, which is found in the cell. This diagram shows the viral infection of the cell via the membrane fusion pathway. The virus binds the ACE2 on the cell receptor, and the virus uses a spike protein in its lipid membrane to bind the ACE2 on the cell surface. And then in the cell membrane is found the enzyme TMPRSS2, which then comes and cleaves the viral spike protein. Further events follow, leading to membrane fusion of the viral membrane with the cell membrane. An opening is created through which the viral RNA enters the cell. This is the endocytic pathway. The virus binds the ACE2 cell surface receptor. The virus uses its spike protein on the lipid bilayer membrane. So spike protein from the virus binds ACE2 on the cell. The whole virus is taken via endocytosis. Here it is the virus in an endosome. An opening is created through which the viral RNA enters the cell. This is the diagram version of the endocytic pathway. Notice the virus attaches the ACE2 via its spike protein. The whole virus is taken by the endosome, and the RNA is then released into the cell. Cathepsin L helps to release viral RNA into the cell, and the cell provides the protease cathepsin L. So protease cathepsin is provided by the cell itself. The membrane fusion pathway. This is the membrane fusion pathway. The virus membrane and the cell membrane fuse, allowing the RNA to enter the cell. This requires TMPRSS2 cleavage at S2 site. Whereas the endocytic pathway, where the virus is taken via endocytosis and found in an endosome in the cell, and then RNA is released. This pathway does not require TMPRSS2. So in this paper, they show that TMPRSS2 is required to infect some of the human airway epithelial cell lines. So this is a cell line culture, a cell line experiment, but they find that in some human airway epithelial cell lines, TMPRSS2 is required for infection. So what they're saying is, if TMPRSS2 is required, then the infection must be by cell fusion pathway, and endocytosis probably is not happening in those cells. So, and this indicates that endocytic pathway may not be working in the lungs, since endocytic pathway does not require TMPRSS2. It does require a furin cleavage. So in some epithelial cell lines, if you knock out the TMPRSS2, virus infection stops, which shows that this infection must be via the cell virus and the cell fusion pathway. It is not endocytosis, since endocytosis does not need TMPRSS2, and if endocytic pathway was working, then even in the absence of TMPRSS2, infection should be taking place. And since it does not in these cell lines, the inference is that in these cell lines at least, the infection is via the cell membrane fusion, with the viral membrane fusion only, and not the endocytic pathway. For infection of a cell, the target cell must provide at least ACE2 and TMPRSS2, transmembrane serine protease 2, and furin, or ACE2 and cathepsin L and furin. This is the spike protein diagram of the spike protein. Notice it is color coded, light blue color and dark purple color. The light blue residues that can change during evolution and so on, whereas the purple ones, there's evolutionary pressure to conserve. But even then, there's the change is very slow in the coronaviruses, especially SARS-CoV-2. It does not change much, as we will see later. And here it is, tucked in is a fusion peptide. We will see this is very important fusion peptide. There is the ACE2 binding site, so we will see later. And this is largely the fusion machinery, that is to say, the fusion of the virus membrane with the cell membrane. This is the area that's involved in carrying out that process, and hence you can see the fusion peptide is tucked in here. We will see the function of that later. And this is the ACE2 binding site. Here it is, the ACE2 binds here. So this largely is the subunit 1 of the S protein, as we will see later, and this is the S2, and S1 has a homology of 64% with SARS-CoV and SARS-CoV-2. So when you look at the SARS-CoV and SARS-CoV-2 sequences, there's a homology of 64% for the S1. Then there's a S2 homology. This is largely this part, as we will see later, has a homology of 91%. So this shows the spike protein. This is the S1, the ACE2 binding site, and here is the S2, and we will see later on this structure in more detail. The thing to note is the SARS-CoV-2 mutation rate or appearance of new strains is low. So it seems SARS-CoV-2 has been with us for about 9 months now, and it has infected a very large population of people, and the rate of appearance of new strains is extremely low, and we will see there's a reason for this. Now we are going to look at the spike protein in more detail. First thing to notice is that spike protein is a trimer. It is made up of 3 polypeptides which are identical, so therefore spike protein is a homotrimer. Here's a structure of a polypeptide that goes to make the trimer. Notice that it has 2 subunits, S1 and S2. This is the S1 subunit involved in binding to the ACE2 receptor on the cell surface. It has a RBD, receptor binding domain. Here's the receptor binding domain. This is the way it binds ACE2 on the cell surface. S2 is more involved in fusion of the 2 membranes, fusion of the viral membrane with the cell membrane, and we will look at this in more detail later. Notice there is a fusion peptide tucked here. There's a fusion peptide, and notice there are 2 cleavage sites, S1, S2 by furin, S2 by TMPRSS2. So it is cleaved at S1, S2 site by furin, and it is also cleaved at S2 site at TMPRSS2. There's a timely organization when and where this cleavage takes place is quite important, and the thing to notice is S2 can mean the subunit 2, or S2 can mean cleavage site 2, which is cleaved by TMPRSS2. So one has to be clear about the context we are using the terminology S2 in. This is just a reminder of what we spoke earlier, that the S1 between SARS, the earlier SARS-CoV and SARS-CoV-2 has a homology of 64%, whereas S2 between the two viruses, SARS-CoV and SARS-CoV-2, has a homology of 91%. So this is the area that has evolved in evolution of the viruses. This area has remained conserved. But even then, take note, the substitution, the rate of change of RNA, the rate of change of viral RNA is very low. Again, reminding that this virus has been around with us for 9 months now, and it has infected a very large number of people, and there have not been that many new strains appearing. So it shows that the viral genome is quite resistant to any evolutionary change. This is a popular diagram showing the receptor binding domain within S1 subunit binding to ACE2, and there is the subunit 2, and here is fusion peptide. Again, a popular diagram showing the sequence of events. Here it is, as the spike protein is produced in the producer cell, as the virus is maturing in the producer cell, S1 S2 cleavage takes place. So S1 S2 cleavage has taken place before the virus leaves its infected cell, and then the virus attaches to the target cell via the target cell's ACE2 receptor and the receptor binding domain of the virus, and then there is a second cleavage by TMPRSS2 at S2 site, which then leads to events leading to cell membrane fusion and infection. Here is the cell infected by the virus, which is producing the virus producer cell, and here it is the target cell to which the virus is going to infect a new cell. So before the virus leaves the infected cell, the producer cell, the spike protein is cleaved at S1 S2 site by furin. So the virus we see in the air or blood of a person and so on, virus that is transmitted from one person to another person, has this S1 S2 cleavage already carried out at spike of the spike protein, and then it attaches to the new target cell and enters that, as we will see in the next slide. If the virus is going to enter the new cell via the endocytic pathway, TMPRSS2 is not required. Nevertheless, cathepsinel and so on will be required. Another protease will be required instead. But the TMPRSS2 is not required if the virus is going to enter the cell via the endocytosis. If the virus is going to enter the cell by membrane fusion pathway, then it requires cleavage at S2 by TMPRSS2. Just a reminder of the terminology: S1, S2 is a cleavage site, S1 is a subunit. So S1 is a subunit, S1, S2 is a cleavage site here, whereas S2 can mean cleavage site, S2 cleavage site, or S2 can mean subunit 2. So here is the sequence of event. The S1, S2 cleavage has already taken place in the producer cell before the virus leaves its infected cell. S1, S2 cleavage has already taken place, and S2 cleavage has not yet taken place. The thing to note is the whole of the trimer is still held intact. So the virus spike protein, the trimer is still intact despite the fact S1, S2 cleavage has taken place. Then when the virus binds a receptor on a new cell, on the target cell, via the receptor binding domain, then there is a S2 cleavage takes place via TMPRSS2. Okay, so once the cleavage at S2 has taken place via TMPRSS2, that is when the subunit 1 comes away from subunit 2. The thing to note it is that the separation takes place at S1 S2 cleavage site. Okay, so although now it is being cleaved at S2 site, but the S1 S2 cleavage site is where the separation takes place. And remember, this cleavage had already taken place before it left the infected cell. So firstly, S1 S2 cleavage has taken place, and if the S2 cleavage has not taken place, the trimer is still intact. Once the S2 cleavage takes place, the S1 comes apart from S2, but at a site where S1 S2 cleavage took place. The thing to note is, after S2 cleavage has taken place, this is now sets in motion a program which enables the cell and virus membrane fusion. This is an experiment comparing the receptor binding domain affinity for ACE2 or the whole spike protein affinity for ACE2. And this is an experiment comparing the two viruses. So when you look at the SARS virus, the earlier coronavirus, the receptor binding domain affinity for ACE2 is relatively low compared to the receptor binding domain affinity to ACE2 of the SARS-CoV-2. When we look at the whole spike protein affinity for ACE2, we find the SARS, the earlier coronavirus, has a relatively high affinity for the whole spike protein, and SARS-CoV-2 has a relatively low affinity for the whole spike protein. We need to remember 2 points. One, isolated receptor binding domain. This is an experimental result, and in patients this situation does not exist. So that is to say, the receptor binding domain is taken out of context, it's taken out of the spike protein, and experiments are carried out. This situation does not happen in the patient. And secondly, SARS-CoV disappeared and reappeared with different affinity of spike protein for ACE2. So one needs to clarify which is the reference strain. This is a reference to a paper discussing the affinity studies on the SARS-CoV and SARS-CoV-2 on their receptor binding domain and the whole spike protein. Now we look at the RNA entry into the cell. Just a reminder of the earlier diagram, the S1, S2 cleavage has taken place before the virus leaves the infected cell. Then it binds ACE2 on a new target cell via the receptor binding domain in the S1 subunit, and then there's a cleavage of the spike protein at S2 site via TMPRSS2, and cell fusion and viral RNA entry takes place. So the events follow, leading to cell fusion and the viral RNA entry into the cell. This is a diagram we developed for the understanding of SARS-CoV, the earlier coronavirus. Nevertheless, I think this is a very good diagram in which to try to develop our understanding of the SARS-CoV-2, the current coronavirus in 2020. This is a spike protein trimer. Here it is, spike protein attached to the virus membrane. There is your S2, S1. Notice S1 cleavage has already, S1, S2 cleavage has already taken place, but the whole protein, the whole complex is still intact. The other thing to notice is the S1 subunit, each one of them can exist in an up or down conformation. Up or down conformation. The down conformation cannot bind ACE2. It's only the ACE2 binding takes place when the S1 subunit is in up conformation. So only when the S1 subunit is in up conformation it can bind ACE2. The other thing to notice, all you need is one of the three subunits, three S1 subunits to be in up conformation for ACE2 binding. You can have more, you can have 2 or 3 which will help binding to ACE2, but one is actually sufficient to achieve that. Here is the more detailed diagram with the spike protein in virus membrane and binding to ACE2. This S2 subunit is here, S1, S2 cleavage has taken place, the whole protein is still intact, whole trimer is still intact. There's one of the subunits, S1 subunits, is actually in the up position, which is bound to ACE2, and ACE2 is in the cell membrane. ACE2 is the cell membrane receptor. Now there's a cleavage at S2, this S2 cleavage site by TMPRSS2. So S2 cleavage takes place and all three subunits, S1 subunits come off. So there it is, S1 subunits come off there. So there's your S2 subunit in the viral membrane, and here's your S2 subunit in the viral membrane. So what is this red bit? Where did that come from? So that red bit is the fusion peptide. That fusion peptide was tucked in there. It is now rearranged itself to expose it at the end of the S2 subunit. So there's been a reorganization of S2 subunit to place fusion peptide in position. So TMPRSS2 has cleaved S2 at S2 cleavage site, and that has got the S1 subunit to come apart and fall off. And also the other thing that has happened is the fusion peptide from within the S2 subunit has now rearranged itself to be exposed at the end. So there it is, the fusion peptide that was tucked in, now it is at the end, and it is also inserted in the cell membrane. Now we come to the question, is there cleavage at all three S1, S2 sites and all three S2 sites in the trimer, or cleavage of just one? I cannot give definite answer, but two issues need consideration: separation of S1 and S2 trimers and exposure of each fusion peptide on each S2 subunit. My assumption is that to achieve both, cleavage is needed at all three S1-2 sites and all three S2 sites. So recapping so far, the TMPRSS2 has cleaved at S2 cleavage site, the S1 subunits have come off, there's the S2 subunit in the virus membrane, there's a rearrangement so that the fusion peptide is now exposed at the end of the S2 subunit, it is now inserted in the cell membrane, host cell membrane, and as the S1 subunits have come off. In the third stage, the part of the S protein nearest to the virus membrane refolds onto a heptide repeat HR1 core to form the 6 helix bundle, 6HB, which is the final post-fusion configuration of the S2 protein. So here it is, there's a folding of the S2 subunit, which is bringing these two membranes together, the virus membrane and the cell membrane. So here it is, the folding is taking place, completely folded onto itself. Now the S2 subunit is completely folded onto itself, and the two membrane, the virus and the cell membranes, have now fused. So the virus and the cell membrane have now fused. An opening opens up so that the viral RNA is now allowed into the cell. So this is the cell fusion pathway. An opening opens up to allow the virus RNA into the cell. So reminder of the complete scheme. So there it is, the virus spike protein attached to the virus membrane, and there is one of the subunits, S1 is in up position, which is bound to S2, and S1 S2 cleavage has already taken place. Nevertheless, the whole spike protein is still intact. Now the TMPRSS2 carries out a cleavage at S2 cleavage site. The S1 subunit and ACE comes off. The S2 subunit is now attached to the virus membrane. There's now a reorganization of the S2 subunit so that the fusion peptide is now exposed at the end. The fusion peptide that was tucked in here is now exposed at the end. This S2 subunit now folds onto itself to bring the two membranes together, to bring the virus and the cell membrane together. So the virus and cell membrane now fuse, an opening is created through which the viral RNA enters the cell. Question remains, how many fusion events are needed? I don't know the answer to that. Nevertheless, what I'm asking is one fusion enough, or do you need two fusion events? That is the same question to ask. Is this fusion enough to open the opening for the viral RNA to enter the cell, or do you need a fusion event here and a fusion event there that will create the opening to allow the viral RNA into the cell? That is a question. In this diagram, two fusion events have taken place to allow the opening to allow the viral RNA into the cell. Here is a complete scheme with the key to particular symbols that shows the complete sequence of events leading to membrane fusion. What is more important, the membrane fusion or the endocytosis pathway? My opinion, and I must emphasize this is my opinion, it needs to be discussed by others in the scientific community, that virus cell fusion pathway is more important than endocytosis. Coronaviruses have invested a lot in virus cell fusion machinery. Subunit 2 is the one that is involved in fusion, membrane fusion, is highly conserved. So cell fusion machinery is highly conserved. Secondly, as we saw earlier, a paper was presented where TMPRSS2 required for virus infection in some lung cell lines. Since endocytosis does not require TMPRSS2, and if TMPRSS2 is obligatory for virus infection in some lung cell lines, it must be going via the cell membrane fusion pathway. So I believe, I am of the opinion at the moment at least, that virus cell fusion pathway is more important than endocytosis. This needs to be discussed by others, and others need to make up their own mind on this issue. Another question is the fate of the remaining S1 on H2. The subunit 1 is actually discarded, but do we get three separate S1 subunits, a trimer of S1, or one single one S1 and a dimer of two S2 subunits? This has implications, and this may have consequences for our patient. When subunit 1 and 2 come apart, we will expose new surfaces, new epitopes to which antibodies can bind. So if the immune response is directed to this part of the spike protein, it is not going to help in neutralizing the virus, because these epitopes are not found on the spike protein found on the virus. On the other hand, we need antibodies to find this part of the spike protein, and this part is common to the discarded subunit 1. So antibodies that are directed against this part, rather than going to bind the whole spike protein to neutralize it, they will in fact bind discarded fragments, which will mop up the antibodies. And in either case, the antibodies binding these new epitopes or antibody binding old epitopes, where the antibodies are directed away from the whole spike protein to the discarded fragment, there will be further pathology, because S1 fragments found bound with antibodies can cause further pathology, for example, immune complex and their deposition in muscles, kidneys, etc., an inflammatory scenario thereon. Does binding of antibody to one subunit stop other subunits from binding ACE2? Do you need to bind all three subunits to stop attachment to the cell? We also need to discuss affinity of the spike protein for the receptor. This is an experiment on the SARS-CoV infectivity. So this is the earlier coronavirus, looking at the proteolytic enzymes trypsin, plasmin, or TMPRSS11A. If you take the vector with the spike protein and incubate with one of the proteolytic enzymes first, and then you add the cells, the target cells, and incubate with the cells, what happens? Or on the other hand, if you take your target cells, incubate with vector with the spike protein first, and then add the proteolytic enzyme, incubate, what does happen? If you incubate the vector with the spike protein with proteolytic enzyme first, and then incubate with the cells, the outcome is reduced infectivity as compared to no proteolytic enzyme. On the other hand, if you incubate cells with the vector spike protein first, and then incubate with the proteolytic enzyme, the outcome is increased infectivity as compared to no proteolytic enzyme. So, if you add the proteolytic enzyme before you add the cells, reduce infectivity. On the other hand, if you incubate cells in vector first, then add the proteolytic enzyme after, there is increased infectivity. This is the graphical representation of the results with the proteolytic enzyme and vector incubated first and then the cells added after. So you notice there's no proteolytic enzyme added at all, and when you add one of the proteolytic enzymes, there's a reduction in infectivity. So in the case of SARS-CoV, the question is there proteolysis of spike protein going on in airways, blood of the SARS-CoV patient? And if yes, does it limit infectivity of the virus, person to person, organ to organ within a person? If the answer is yes, then the question becomes, has SARS-CoV-2 overcome this limitation by proteolysis and become more easily transmissible? This is a brief diagram to show the relationship of the alveolar type 2 cell in the alveolar. Notice that the alveolar type 2 cells are here producing surfactant under normal circumstances. So notice on the alveolar type 2 cell has actually a asymmetrical membrane, that is, say this is the basolateral membrane and this is the apical membrane facing the alveolar air space. ACE2 expression is high on the apical membrane. That's where the ACE2 expression is most found. And this is also on the apical membrane where the SARS-CoV and SARS-CoV-2, in fact, they enter the cell through here and also leave the cell here going towards the air. Now we come to the viral RNA synthesis. The virus brings its own RNA for translation for protein synthesis. Transfer RNA and ribosomal RNA are hijacked from the host cell, so the virus uses cell's protein synthesis machinery to synthesize its own proteins. Reminder of some basic information on RNA. Peptide synthesis starts at the first AUG codon from the 5' end. Peptide synthesis stops at the first codon UAA, UAG, or UGA. But there's a but, as we will see. The newly synthesized RNA strand is synthesized from 5' to 3' direction. Humans can only synthesize RNA using DNA as template. Enzyme equals DNA-directed RNA polymerase. We cannot synthesize RNA using RNA as template. We lack RNA-dependent RNA polymerase. So the virus has to bring its own RNA-dependent RNA polymerase. Messenger RNA has 5' capping and untranslated poly A tails at a 3' end. Synthesis of RNA is from 5' to 3' end. SARS-CoV-2 RNA may be divided into two major groupings. One is coding for the non-structural proteins, the other is for the coding of structural proteins. Structural proteins are the scaffolding proteins of the virus. One has to be a bit careful because the structural proteins may have more than one function, hence may also be acting as non-structural proteins. So this region of the RNA will code for the non-structural proteins. Peptide 1A and peptide 1AB are synthesized. Notice within each of these polypeptides there are several proteins, so these are multi-protein polypeptides which will be cleaved to produce individual proteins. This is a larger diagram showing polypeptide 1A being synthesized and polypeptide 1AB being synthesized, but note the viral RNA has a stop codon here and a stop codon here. In AUG, this is where the synthesis of both of these proteins starts. So, virus starts its protein synthesis here, translation here at the first AUG codon, carries on, it comes to the stop codon, and normally it should stop synthesizing the protein here. And sometimes it does, but not always. Sometimes it carries on synthesis. What happens here is there's a programmed -1 ribosomal frame shift to control the relative expression of their proteins. So, peptide synthesis stops at the first codon to produce polypeptide 1A, as we saw. But sometimes there is a slippage and ribosomal machinery carries on synthesizing longer polypeptides up to the second stop codon. At the first stop codon, slippage also involves frame shift. So either protein synthesis will stop here or it will carry on through to the next stop codon with a -1 ribosomal frame shift. At the second stop codon, polypeptide synthesis is terminated definitely, no ifs or buts. So up here when the polypeptide synthesis is stopped definitely. Polypeptide B is only synthesized as continuation of 1A to make 1AB. So you cannot synthesize this portion by itself. It can only be synthesized as part of this larger polypeptide. This is a summary diagram to show the polypeptide 1A being synthesized up to the stop codon here, 1AB synthesized by -1 ribosomal frame shift and slippage, the machinery going on to continue synthesis of the protein and stopping here. And notice this part of the polypeptide can only be synthesized as part of 1A. You cannot have this part being synthesized by itself. The other thing to notice is this RNA has a leader sequence and a TRSL, transcription regulatory sequence L. Remember this, and this is quite an important sequence, as you will see in the TRSL. This is a reference to the ribosomal -1 frame shift. This is the open reading frame 1A coding for the polypeptide 1A, and open reading frame 1B, which together with open reading frame 1A, it codes for the polypeptide 1AB. Not shown here is the leader sequence or the transcription regulatory sequence, LTRSL. This is the coding sequence, and these are the individual proteins contained within the polypeptides. Notice the PL-PRO is the papain-like proteinase, 3CL-PRO is the 3-chymotrypsin-like protease, and we will see that these polypeptides are cleaved into the individual proteins, and RDRP is the RNA, virally coded RNA, directed RNA polymerase. Just want to give an example here. This protein here, exon N and N7 methyltransferase. So notice that same protein has two different functions, exonuclease and N7 methyltransferase. You see the polypeptide 1A and polypeptide 1AB synthesized. The papain-like proteinase and 3-chymotrypsin-like protease, they can function as proteases while they're still within the peptide. So there are several proteins included on each polypeptide, and this proteinase, papain-like proteinase and 3-chymotrypsin-like protease, they can function as proteases and cleave themselves out of this peptide. Once they've cleaved themselves out, they can now go on to cleave the rest of the sites to create several proteins from this polypeptide. So in this way, the peptides 1A and peptides 1AB are cleaved to produce several different types of non-structural proteins. I have put together this slide, tried to put in perspective the proteases. So there are proteases provided by the cell, the TMPRSS2, Furin, and cathepsin maybe for the endocytic pathway, and proteins that are virally coded, the 3-chymotrypsin-like protease and papain-like proteinase. So here this is the open reading frame 1A and 1B producing the polypeptide 1A or polypeptide 1AB, and the proteins that are produced are grouped into their major grouping. So we've got the host modulation proteins here, polyprotein cleavage, and double membrane vesicle formation here, in the RNA transcription and replication proteins here. Notice the double membrane vesicle formation. This is quite an important structure I shall be dealing with in the next few slides. The point here is that these double membrane vesicles are not normal cell structures. They are induced by the virus, so that the virus induces the cell to make these double membrane vesicles, which we'll discuss in the next few slides. This is the reference paper for the double membrane vesicles, and notice this is from SARS-CoV, this particular electron micrograph. It is a clinical specimen from SARS-CoV patient. Double membrane vesicles are not normal structures found in a cell. SARS-CoV-2 induces the cell to synthesize these double membrane vesicles. Appreciate this electron micrograph is of a infection by SARS-CoV, infection of the cell by SARS-CoV, but it's reasonable to assume that coronaviruses induce these double membrane vesicles, and it is therefore reasonable to assume that SARS-CoV-2 also induces the cell to synthesize these double membrane vesicles. All RNA replication takes place within the double membrane vesicles, hence double-stranded RNA is found only within double membrane vesicles. In fact, we can clearly see that there is a granular material inside the double membrane vesicles that actually is the RNA, and this is not to be confused with the virus particles. You do not find virus particles in double membrane vesicles. Double membrane vesicles do not contain virus particles, they do contain the viral RNA. There are many hundreds of these double membrane vesicles in an infected cell. So if you look at it, this is the cross section showing how many double membrane vesicles there are. Now imagine these in a three-dimensional space in the cell. This gives an idea of some kind of magnitude of these number of these double membrane vesicles. Double membrane vesicles do not contain mature virus particles. Repeat, double membrane vesicles do not contain mature virus particles. Mature virus particles are assembled later in the endoplasmic reticulum or the Golgi. Double membrane vesicles is where viral RNA synthesis takes place. These provide a place for the double-stranded RNA intermediates to hide from cells' defense systems. Double-stranded RNA is not a normal cell constituent. Cells only make DNA to RNA. Cells do not make RNA to RNA. Cell has innate immune mechanisms to identify as foreign intruder, attacking and putting in response to limit the infection. Double-stranded RNA is detected as foreign material. It is thought the double-stranded RNA is hidden from cell defense mechanisms inside the double membrane vesicles. Remember we discussed earlier the lipid bilayer. The double membrane vesicles have two layers of lipid bilayer. So there's a lipid bilayer here, goes all the way around, goes all the way around. So it's a double membrane vesicles are made up of two lipid bilayers. This is an electron micrograph of SARS-CoV in culture cells, ultrastructure of alveolar type 2 cells infected with SARS-CoV. Cells were cultured, as it says here. So here we can see virus particles in secretory vesicles here, virus particles being extruded, and here the virus particles being extruded here, and here the virus particles in secretory vesicles again. And here is the double membrane vesicle. It does not contain any virus particles. And notice here at higher magnification here, double membrane vesicles do not contain any virus particles. This is a cells infected with SARS-CoV virus, the earlier coronavirus. How much of cells and whole patients' resources are being diverted to make this DMV, in addition to other things that are going on? How much of the hypoxic patient's oxygen supply is being used to synthesize this DMV? So recap, the non-structural proteins are made. A polypeptide 1A and 1AB is synthesized containing several proteins each, and there's a proteolysis step that cleaves these polypeptides to produce individual proteins. So individual proteins are produced by cleavage of the polypeptide 1A or 1B. Proteolysis of polypeptide 1A or 1AB will produce these non-structural proteins. Some of these non-structural proteins are involved in RNA synthesis. Replicase transcriptase complex is formed by some of these enzymes that will carry out RNA synthesis, and these proteins combine with the RNA itself. The same molecule of RNA that the virus brought with it now forms a replicase transcriptase complex. This then moves into the double membrane vesicles. These double membrane vesicles are also induced to be synthesized from the cell by some of these non-structural proteins. So there are non-structural proteins that cause the cell to induce the cell to synthesize double membrane vesicles, and this then moves into, so this replicase transcriptase complex then moves into the double membrane vesicles. In the double membrane vesicle, RNA synthesis is carried out. Notice minus RNA strands are made from 5' to 3' direction, and notice there are several different lengths of RNA being made. Not all of them are shown here, but there are several different lengths of RNA being made. This diagram showing the replicase transcription complex moving into the DMV. Different lengths of negative strand RNA are made using the positive strand of RNA as a template, and note, as we said earlier, different lengths of RNA are made. So there are different types of molecules made of our negative strand of RNA which are different lengths. Then each one of those minor strand of RNA, the different length of RNA, is used to make different lengths of positive strand RNA. So each one of the negative strand RNA synthesized is copied, is then used to make positive strand of RNA. The positive strand RNA leaves the DMV and enter the cytoplasm. The minus RNA stays within the double membrane vesicle. Just a reminder of the correct sequence. From the positive strand, different lengths of minus RNA are made, and each one of those minus RNA is then copied into plus RNA. This is the wrong sequence. You do not get one minus RNA from which different lengths of positive strands are made. So this is the wrong sequence, this is the correct sequence. The minus RNA does not leave the double membrane vesicle. So the minus RNA stays in the DMV. Recap, we have now created different lengths of RNAs which will be used to make proteins, and also we have created copies of the whole length RNA which will be used to package RNA as a genetic material for the new virus particles made. Now we are going to look at the synthesis of the structural proteins. The proteins are going to be used to make the scaffolding of the virus. We are looking at this group of proteins now, the structural proteins, and how these are synthesized. SARS-CoV-2 has created these different lengths of RNAs coded by the sequence for these structural proteins here. These are effectively analogous to mRNAs, that is, they have 5' capping and 3' untranslated poly A tails. And notice, so these are different proteins coded by the virus, and there is a start codon, AUG, and a stop codon. For this protein, spike protein, there is another start codon and stop codon. For this protein, start codon and stop codon for this protein, and we'll see the significance of this in the next few slides. We look at this RNA molecule to start with. It has a coding sequence for the spike protein with a start codon and a stop codon. The next sequence downstream is coding sequence for 3A with a start codon and a stop codon, and so on. The next species of RNA, we see it has the coding sequence for the spike protein missing. So it starts at the coding sequence for the 3A with a start codon and a stop codon. E protein is the next one with a start codon and a stop codon. And the next RNA has coding sequence for the spike protein and 3A missing, and it starts at the envelope protein with a start codon and a stop codon, and so on. So this way we carry on. Each time another protein sequence is taken out. So up here the spike protein coding sequence is missing, here the spike protein and 3A coding sequence is missing, and so on. We'll start looking at this RNA molecule with a coding sequence for the spike protein at its 5' end with the AUG start codon and a stop codon, and other proteins downstream. So what happens is protein synthesis starts at this AUG codon and for this using this RNA molecule, and it stops when it reaches this stop codon, and the rest of the RNA is wasted. Next we look at this molecule of RNA with a protein coding sequence for 3A at its 5' end with a start codon and a stop codon. So the protein synthesis starts at this start codon AUG, and when it reaches the stop codon it stops, and rest of the RNA is wasted. So here RNA with the S protein at its 5' end is used. The S protein is made, synthesis stops at the first stop codon and counted. Rest of the RNA is wasted. So to make 3A protein, RNA with 3A protein at its 5' end is used. 3A protein is made, synthesis stops at the first stop codon and counted, and rest of the RNA is wasted. Again, this virus can plunder so much of the cell's person's resources to get what it wants. It's making such of the RNA it does not use. Now we look at the mechanism how these different lengths of RNA were created in the first place. Note the organization of SARS-CoV-2 RNA. Note there's a leader sequence, and there's a TRSL, which is the transcription regulation sequence L, and there's the TRSB, the transcription regulation sequence B. So in front of each one of these proteins that are structural proteins that are going to be synthesized, there's a TRSB sequence. So there's TRSB sequence at 5' end of the spike protein, TRSB sequence at 3A, and so on. Remember, there are no TRSB sequences in the open reading frame A, the coding sequence for the non-structural proteins. This is the enzyme complex which is responsible for synthesis of the RNA. So it will synthesize the new minus strand RNA first, then from the minus strand RNA plus strand RNA. Notice the two key players, they're all important enzyme components of this enzyme, but NSP12 is the RNA-directed RNA polymerase, and there is the NSP14, which is the exonuclease. For synthesis of the negative strand of the RNA, whole of the molecule containing whole of the viral genome is used, and the synthesis starts at from the 3' end of the RNA molecule to be copied, and note that the synthesis of new RNA is made at 5' end and continues towards the 3' end. So it starts synthesizing here. What happens is, when it reaches one of the TRSBs, transcription regulation sequence B, a choice is made whether to just continue making RNA as normal or jump. Somehow, when it has a decision making, it can either go past its TRSB or jump up to the TRSL and continue synthesis there. So this red dotted line represents the RNA sequence that is not copied. It's been jumped over, if you like, and gone straight to TRS. So 5' RNA synthesis starts here, it reaches its TRSB sequence, and here then it just jumps over the rest of the RNA and goes straight to the TRSL and finishes RNA synthesis. So an RNA sequence is created, a negative strand RNA sequence is created for this sequence, which can code, the positive strand can code for these proteins, this sequence here. So there is from this negative RNA, positive strand RNA strand is made. So this is a diagram taken from this reference here, and notice that different length RNA sequences are being made. The machine reaches a TRSB, transcription regulation sequence B, and a choice is made whether to carry on to the next TRSB or jump over and go straight to the TRSL and continue synthesis there. So from the negative strand, positive strand is then copied. In this way, subgenomic RNAs are made, minus. They contain a certain length of sequence, and then the leader sequence with TRSL is at the end of the RNA. The RNA is capped. I am guessing, but I would guess that there will be some kind of looping mechanism going on. This RNA will form a loop to bring these sequences close together, so the RNA desynthesizing machinery, the complex, can carry on onto the TRSL. I don't think there's actually a jump. Anything is possible, but what I would guess, it's not likely that there's a jump. It is more likely that there's a loop formation that brings these two sequences close together, so the machinery can carry on. And again, a choice is made at each TRSB whether to go on to straight onto this TRSL or carry on to the next TRSB. It's worth saying this area is being worked on now with SARS-CoV-2. I would be a bit concerned that this SARS-CoV happened about 20 years ago, and this work should have been carried on after SARS-CoV. We should not have given up the amount of concern this virus caused. We should have predicted that this virus could evolve into something like what it has, and we should have been working on this kind of science behind the SARS-CoV all these 20 years. It's a shame that this took off when the SARS-CoV-2 came in and caused so much damage. We should have been prepared for it. We should have anticipated it. This is just a suggestion. It may or may not be right. It's got to be taken with a pinch of salt. But the idea suggested is that there's some kind of looping mechanism that brings these two sequences together so that the RDRP complex can actually jump over from this B sequence to this L sequence and finish off the RNA synthesis. Recap: the virus non-structural proteins are used to induce the cell to make these double membrane vesicles. The viral RNA molecule combines with the replication transcriptase complex, the enzyme is responsible for viral RNA synthesis. This then moves into the double membrane vesicle. So this represents the double membrane vesicle, and there the transcription takes place. Minus RNAs are synthesized. Different lengths of minus RNAs are synthesized by the mechanism we just discussed, and then within the DMV, each one of these lengths of RNA, minus RNA, is copied to make a plus RNA. These are the ones that are going to be used for the protein synthesis, and there is a whole genomic RNA made that will be used as a genome for the new virus. So the plus strands of the RNAs come out, remembering that minus strands do not leave the DMV, they stay within the double membrane vesicle. This is the enzyme complex responsible for synthesis of the new strand of RNA, and one of the key players in this complex is the RNA-dependent RNA polymerase. The thing to remember about this enzyme is that it is quite error-prone. It will create lots of wrongful additions in the RNA that is being synthesized, wrong substitutions, and effectively mutations. But the SARS-CoV-2 has been around for nearly a year. It has infected a large number of people, and mutations in the new strains in this virus are very few. So it's resisting change. And one of the things is that it doesn't need to resist this change because this RNA molecule is very large, and by chance there will be lots of mutations, and total sum of these lots of mutations would be lethal for the virus, and the virus will not be able to survive for long because of the mutation it carries by chance, and especially since the RNA dependent RNA polymerase is quite error prone anyway. So this exonuclease detects errors and removes them. So virus has carries with it RNA error checking machinery that locates the errors and removes them. So this is stopping the virus from evolving. Hopefully it will help us because it will not be able to evade vaccine or drugs by developing resistance. On the other hand, the drugs, some of the drugs against these viruses work by creating mutations in the virus, and Axon will be resisting that. Drugs will not work very effectively. The drugs that cause mutations in the viral RNA will not work very effectively because exonuclease is there removing in any mutations. So here's an experiment reference to this paper whereby exon and mutants have been created, and notice that in the wild type the total number of substitutions per genome is low, but in the mutated exon and variants they do accumulate a lot of substitutions per genome. So showing that SARS-CoV exon mutant viruses accumulate nucleotide substitutions across the genome. So this is a SARS-CoV experiment. Exonuclease N removes errors. So this is the exonuclease N, which will limit wrong nucleotide incorporation into the RNA, and this may be a good thing for us in some respects because exonuclease N is also limiting the virus from developing new strains. So hopefully when we have a vaccine, the exonuclease N, the enzyme of the virus itself, will limit the virus from developing new resistant strains to the vaccine, or when we have some drugs available against this virus, hopefully, which we will have soon, then it is the exonuclease N that will be limiting the virus from developing drug-resistant strains. On the other hand, exonuclease N is also hindering the development of drugs which work by mutating the virus. So there are many drugs against RNA viruses that will cause mutations of the RNA, therefore stopping the virus from replicating successfully. So exonuclease N is a good thing in some ways. It is limiting the development of drug-resistant or vaccine-resistant strains, but it is also hindering the development of drugs which work by mutating the virus. Now we come to summarize what has been going on. The virus, via its spike protein attached to the receptor, the ACE2 on the cell membrane, TMPRSS2 in the cell membrane provided by the cell, cleaves the spike protein, leading to events that led to the membrane fusion, the fusion of the viral membrane with the cell membrane. An opening was created to allow the RNA of the virus to enter the cell. The same molecule of the RNA that virus brought was used to translate into one of the two polypeptides, polypeptide 1A and polypeptide 1B. Each of these polypeptides contains several proteins, which by proteolysis individual proteins were made. Some of these individual proteins were used for the RNA synthesis machinery. These combined with the RNA molecule, the same molecule that the virus brought, which then moved into the double membrane vesicles. These double membrane vesicles were induced in the cell by some of the other proteins which are required to induction of double membrane vesicles from the cell. These double membrane vesicles are not normal structure in the cell, they are induced by the virus, and this RNA inside the double membrane vesicles was then copied to make monostrand RNA, whole length monostrand RNA plus several shorter length monostrand RNAs. All these monostrand RNAs, so shorter length and the whole length, were then copied to make plus strand RNA. So the whole length RNA plus strand RNA will be used for packaging into the new virion, whereas the shorter length RNAs were used to translate certain proteins, the spike protein, envelope protein, membrane glycoprotein, and N protein. The N protein and the whole length RNA combine together to make the nucleocapsid, which moves into the ERGIC, and then the other structural proteins via the endoplasmic reticulum also move into the ERGIC, the endoplasmic reticulum Golgi intermediate complex. This then moves into the Golgi apparatus to form secretive vesicles where the virus is assembled fully, and there it will be cleaved by the furin to actually be ready to be given out to the outside world by exocytosis. So this slide shows the translation of the structural protein which end up in the ER, and translation of the N protein which combines with the RNA. Notice that the plus RNA came out of the double membrane vesicle into the cytoplasm, so it is in the cytoplasm that the synthesis of the structural protein is taking place, and probably in the endoplasmic reticulum that's where synthesis is taking place. In any case, these proteins do end up in the endoplasmic reticulum, which then moves into the endoplasmic reticulum Golgi intermediate complex with the N nucleocapsid. Nucleocapsid is made up of the N protein and the whole length RNA, genomic RNA. So whole length genomic RNA and the N protein make up the nucleocapsid, which moves into the ERGIC along with structural proteins ready for the whole virus to be assembled. So the whole virus is assembled in the ERGIC. The virus assembled in the vesicles, and then cleavage of the spike protein at S1, S2 by furin takes place, ready for maturation and exit to the outside world by exocytosis. This is a beautiful picture of the horrible beast that is now coming out, ready to infect many other people. Notice so many virus particles are coming out from one cell, and there may be other sites in the cell where these virus particles are coming out. So you can imagine the viral load the patient is being subjected to, how many virus particles are coming out just in this place, and when you compare that there may be other sites in this cell where the virus particles are coming out, then there are so many other cells that are infected. This is an immense viral load the patient is having to face.